I'd like to welcome Trevor and all of you to the live question and answer session of the seminar. Please ask your questions on the box on the screen and we'll ask the most popular questions. So please use the upvoting function. Thank you so much, Trevor, for that great talk. And my question is, has the baby biome study uncovered any longer term impact on childhood health between the vaginal C-section and C-section babies or the breastfed babies? Have you tracked the longer term health outcomes?
No, that is the goal. And I think it's the type of science we're doing now, this long-term science. I think it's one reason why, I think we're fortunate to have the Sanger behind this long-term funding. But we need longer-term sampling. And some of these phenotypes we're interested, these clinical phenotypes, obesity, asthma, they start to present themselves at 5 years. So we haven't gotten there yet, but this is we're at the beginning of this road now to link these together.
Thank you.
Okay. And just to follow up a bit on that, I mean, I guess in terms of longitudinal, ideally you would follow these people for the rest of their lives, their adult lives. But yeah, you mentioned 5 as a critical stage or age. Are there other kind of developmental stages that might be particularly important to follow this cohort through?
Yes, it's usually what we power the study to focus on, right? So we're involved in some, the change study I mentioned towards the end. And this is looking at anything from neurodevelopment to mortality. So we get these endpoints, but those are the key stages. So developmentally, the cognition, the first year or two of life is extremely important. So we can map these, and that's the goal, to see how the bugs and the early composition of the microbiome can impact that.
Great. So I'm just going to ask one of the top rated question here that comes from Vivek. So the question is, did you perform any analysis linking the changing microbiome to changing gene expression? I think that's in the host, and how these interact. And then he mentions that in his undergrad, they had experiments in mice with stress, have varied gene expression within 2 weeks and a varied gut microbiome. So it'd be interesting to translate this to humans.
Yeah, definitely in humans, we're not there. In mice, we do look, and we have some experiments. I didn't discuss today. We're looking at the impact of these bugs in mice with certain diets and how they impact the development of the immune system, how they can resist pathogens. So we're starting to mimic this, what we see in children in mice, but ultimately we want to use this as a basis to go into humans, and usually need this type of data to justify that. There's another way of looking at the host. I mean, the expression is one thing, but also the genetics. I think there's going to be some very important genes. FUT2 is one that is pretty, important to the lab. We focus, this puts fucose in the mucus of it, and then this feeds the bugs. And so there's another way to approach the host interaction in addition to expression, but also the genetics. So these are what we're looking forward to.
Okay, great. Thank you, Trevor.
Thank you. And this is quite a wide-ranging question, Trevor, but how does the gut microbiome vary by geography, diet and ethnicity? Do you see changes in the gut microbiome according to geography or diet or ethnicity? And what is one of these a key driver of the variation you see?
Okay. There's a lot of things there. Diet.
Yeah. Diet, yes. Start with diet and geography, for example. Start with diet.
The geography question, I think, yes, there is evidence that there's certain microbes and it's linked to lifestyle as well. And where you live in urban versus rural, as hunter-gatherers, still, these microbiomes have been profiled and they're different based on that, but it's the diet as well that comes into play. No, we have the data to answer this. In our I talked about the baby biome phase 2. We'll have these data to look at, especially breastfeeding, the diet, to understand how they intersect. But I don't know if that answers the question clearly. We're not quite there, but.
Because you mentioned, Trevor, that there's a large bias in the sequencing of the microbiome, and that's predominantly around, I think, European and North American, and I anticipate it would also be around Caucasians as well. So I mean, how much do we really know? I mean, you kind of intuitively you think there will be a lot of variation. Have we really even begun to ask the question? Do we have any early insights?
I guess that could, are we mapping the collective human microbiome is another way to phrase that. Do we have references? This is one of our big goals, not just with our partners. I mentioned this towards the end. You can see the discrepancy in it's uneven in what we focused on. Yes, it's North American, Europe, and Asia, mainly China. And we're going through Latin Biota. We just had a very, very exciting Latin Biota 1 meeting last week. And you can see this, it's on the internet, you can watch it. And Gregorio is an international fellow at the Sanger, presents the Latin Biota. And so this is sampling people from different lifestyles and cultures and different diets across Latin America. So we'll have to do this in phases, but yeah, I believe we will sequence and map the collective human microbiome, certainly.
Okay. And kind of an extension of that question, Julia's question, and I'll make it my next question is are there efforts to actually generate databases that begin to aggregate this data and consolidate it into one place, much as, increasingly things like cancer genome sequencing has been aggregated into large-scale databases?
Yeah, there are. I don't know if it's properly consolidated in one place. I mean, we work closely with EBI. They're on campus with us, Rob Finn, and the Magnify website. This is a portal we use to share data in a standardized way. It's probably one of the more common and very high standards, but I do think there is a need to link these. And the data in Latin America, for example, how are we going to handle that and share that and link it together? These are sort of questions we're starting to ask now. But I do believe using your cancer program as an example, those are standards we should aim for, certainly.
Thank you.
Thank you. So Trevor, there's lots of sort of direct to consumer products that claim to give you gut health. And what's your opinion of these sort of non-prescription commercial products that could aim to give you a better gut biome?
Well, I mean, there are products out there. If you look at using microbes in a beneficial way, I think there are two ways to look at. There are nutraceuticals. I mean, the probiotics is something you can buy at the local grocery store or pharmacy, and then there's pharmaceuticals, things that have to a doctor needs to prescribe to you, and those have to undergo robust, strict clinical studies to look for safety and efficacy. So there are different paths, and the data, the science behind the pharmaceutical approach is going to be strong, and it's coming. This idea of live bacterial therapeutics exists. If it works for you, take it. But I think there's questions around engraftment, and are they the appropriate strains that still exist? So if it works for you, take it. But don't be disappointed if it doesn't work, I guess.
Okay. Right.
Yes, I was going to say, because there's such interest in this area, it's obviously an area where commercial companies will want to be involved and promoting things, whereas the pharmaceuticals need to take may take longer to reach the market potentially. So it's just
there's some really cool, this idea of live bacterial therapeutics. Of course, I'm a co-founder of a spin-off from the Sanger Microbiotica, and we're involved in this industry. And there's some very cool, important clinical readouts from our other companies in America around this, using single bugs or combinations of bugs to deal with allergies and autoimmune diseases. So those are the pathways. This has been a decade of clinical trials and back and forth. And so it's coming. It is coming in this industry.
That's great. Thank you.
Okay, great. So Trevor, the next question comes from Yaza. So they ask, do you plan to add metabolomics into your studies in order to apply function to the baby biome models, as it's more important of what these bugs are doing than their presence?
Well, I don't know that last point, if I agree. I mean, you need their presence to be doing, so I'm not sure if I follow that logic, but I get your point. I think taxonomy, the presence of the bug and the functions are equally important. They're there together, and it's just a deeper understanding. I think by identifying the species, the composition is a good starting point, and to look at pathways. And I do know in our group, we've looked at a lot of these bugs at the metagenomic level and individual bugs, and we can find pathways that are very important for carbohydrate metabolism. And this allows us to develop a biological hypothesis we can use the bugs to address and do these very experiments. And now we're setting up systems now to grow these bugs in a high throughput manner to screen them for certain activities as well, using RNA-seq and other genomic technologies. But yes, this is the next, and I believe it's the next phase for human microbiome science to get into causation, exactly what you're discussing there.
Yeah. And you know, I mean, you talk a little bit about the application of, well, therapeutics, you know, having bugs or combination of bugs, but I guess, do you actually ultimately see moving beyond adding the bugs, but actually just adding back the metabolites or whatever it is that these actual bugs are doing to the cells, as opposed to actually adding the bugs themselves? Because presumably from a kind of therapeutic perspective, from a manufacturing perspective, there would be lots of advantages of that.
Yeah, yes, it is happening. I believe all these things are being explored in the human microbiome sciences space. And to some degree, the small molecule approach is what big pharma is well suited to do. So you can see a draw and almost a gravity towards that. And other companies think about using the bugs as drugs, is what, you know, it's we sometimes say is there. But yeah, these approaches will be there. And I bet you we're going to find amazing things from these bugs. They're so co-evolved with humans and linked to our development and resistant to diseases. And we just got to learn how to grow the bugs in a very controlled way. And think of them as microbial machines, in a way, and get them. This is long-term vision, I believe.
Thank you.
So, sorry, I could follow up that question in quite a few different ways. So I'm going to pick up on one of one of the elements that clearly there's an epidemiological link between the microbiome and various diseases, and that's increasingly clear with robust evidence. How do you then go about establishing causality when you're dealing with such complex biological system? Because you've got the bugs, you've got the host, you've got the environment. Can you link causality, or is it just establishing links?
I mean, often this is a big question, asked big. And sometimes you break it down. And so, you know, there's different ways. I'll answer this a couple of ways. So one way to break it down, we have a deep focus on maternal transmission now. So this is the earliest stage, right? And we can look at this with our sampling and ask questions and understand that aspect. And then we can ask how those early colonizers, pioneering bugs, impact other things like the assembly of the microbiome. What we're finding is those interactions, positive interactions, probably driven through co-metabolism, not really stick them, but keep them together and they grow. And so you can imagine breaking it down, asking individual questions, but ultimately we discover we make these observations in humans, in children and in moms. And so to go full circle, to do this properly, you have to do the experiments in humans and do interventional studies. And this is where we'd like to go with our study, with this work, and also within microbiotica. That's the ultimate question: do they work in humans? You have to. Unfortunately, I had to form a company to find out, but you know that is the big driver.
Yeah, that's fascinating.
And Trevor, one thing just about the baby microbiome. You talk about the differences in the composition of the microbiome between a C-section and a vaginal birth. I'm aware, I'm no expert about this, that in some cases when a baby is born through cesarean section, there's actually a vaginal swab taken and then it's kind of inoculated the baby.
Yes.
Is that something you've looked at? Is that supported through your evidence in terms of colonizing that baby very rapidly?
It's being done, and we friends of ours in New York City have done. They published a Nature Medicine paper. It does impact the early colonization, which is expected. These are bugs that could colonize. If I believe correctly, I don't think they linked it to any change in the bacterial, the baby's phenotype or the growth. But it has been done and established you can engraft the bugs. Are we doing it? I'm not sure. Or maybe our analysis points to different types of bugs. So they're the vaginal bugs. Our analysis pointing towards gut bugs. Those are the bugs. So I think, and we're approaching this in different ways. So we'll find out, which is right. But it's exciting to know that so much attention is being put into this. Yeah. I think there will be good candidates.
But presumably, this could actually suppress the pathogenic bugs or those strains. That would be the one.
I think it's hypothesis. One of the most important functions, right? So you're so vulnerable. You're there's this huge petri dish when you come out of your mom, and the bugs are in, and you need those defensive breast milk. I mentioned this as well in the lecture. And there's antimicrobial properties in breast milk in addition to food for the bugs. And so you're very vulnerable in this period. So it's probably one of the main functions in addition to metabolism.
Thank you.
That's great. Trevor, just another huge question. Are there microbiome seems to be associated with allergy? You said allergies, cancers. Are there any diseases where microbiome isn't being linked, or do you think this is just a neglected area of research and medicine until now, and suddenly we're going to open this vast new palace of information?
Yeah, it seems to be quite. I'm not sure I can say it impacts all diseases. I think that's a bit stretched, but it impacts our physiology. Like I said in the beginning, we are an ecosystem, and it's not us and them. We're together, and we have to learn to live in harmony, right? And there's long-term views part of this. So it impacts our physiology. I mentioned there's so many metabolites and chemicals in our blood that come from the bugs in our guts, and so I think they're always involved to some degree. It could be a tiny bit. It could be a massive amount. So I think of it that way.
Yeah, that's interesting.
Trevor, just probably squeeze in one last question before we have to conclude. When you do these types of studies, there's often, enormous kind of technical hurdles. And I just wondered if you could reflect for a minute on, how does one go from a stool sample to a inventory of a human's microbiome? And there must be enormous challenges in terms of sample preparation and analysis. And I wonder if you could kind of comment on that.
Yeah, and that's part of our current goal in the lab, and this QQ is to scale this up and optimize it. And so there are technical challenges around it. How you biobank them and store them in a very consistent way. And so we're looking at clever ways and moving to reagents to preserve it and stabilize it right at source. So as we go into low income countries, there are different challenges there. -80 is not always there available to you. So that's our plan, is to just open it up to drive the price of edited genomes down significantly so other people can access this. So yeah, this is, I could go through my SOP book, but there's been tremendous through the Sanger lab in the last decade, just shrinking down, optimizing aspects, and we want to be able to spread and democratize this approach. And so the analytics itself, it's, the first stage of the way we approach it is to get the list of bugs, which you can flip across the feed functions. We talked about that a minute ago. And then the analytics beyond that is more bespoke to the question. So I think we're laying this out, and I mentioned this a bit in the lecture, to democratize microbiome sciences.
I think it's now 5 o'clock, so I think we're going to conclude the question and answer session. But I'd like to thank Trevor Lawley once again for his presentation and joining us for the question and answer, as well as the audience who's joining us live and later through the recorded sessions. Just to point out that we have another seminar lined up for December. This will be given by Dr. Maz Hanifa, who's going to talk about her work on the Human Cell Atlas. So that's on the 15th of December.